Hi guys, this is Mrs. Foy, and this is part 2 of the AP Biology Immune System lecture. So when I last was with you guys, we talked about the barrier defenses, and then we talked about the inflammatory response, which is part of the innate immune system reaction. And then we gave just a little bit of an introduction to the acquired or specific or the adaptive immune system as the crowning achievement of vertebrate evolution. So today we're going to talk about that. And then we're also going to talk about a virus that exploits the acquired immune system. So we'll talk about that. So let's take just a look in general about what happens in the acquired immune system. So we have an antigen. We have some type of antigen that is on a pathogen that is infecting us, all right? And so what happens is that is engulfed by an antigen presenting cell. That can be a dendritic cell, or it can be a macrophage, or it can be a B cell. And what happens is that antigen presenting cell then goes to dock with the helper T cell. And we'll see it basically says, here, helper T cell, this is a piece of the invader that's infecting us. What are you going to do about it? And so the helper T cell then begins to orchestrate or conduct the entire adaptive immune system response. And it will activate the cytotoxic T cells, which are also called killer T cells. It will also activate B cells to do their things. And the B cells themselves can also be antigen presenting cells, but we'll talk more about that later. So then in the humors, in the blood and in the lymph system, these B cells are going to be activated, and they are going to basically split into two different populations. One population of cells we call the plasma cells. And the plasma cells we're going to see are going to secrete antibodies. Think of antibodies as protein handcuffs. That's basically what they do. They bond to the antigens on the invading cell, and they kind of immobilize it, just like a handcuff would. Some of the other B cells differentiate and become memory B cells. And these create a pile of cells that have kind of the blueprint of that invading cell's antigens and are ready to respond with some more antibodies when it comes back again. So now let's talk about these helper T cells. The helper T cells also can develop into memory helper T cells with the memory of that exposure from the original antigen presenting cell. And they can also help to activate the memory B cells and the memory cytotoxic T cells, those memory T cell, cytotoxic or killer T cells, when there is a second exposure to of the antigen. Meanwhile, these cytotoxic T cells become activated, and they undergo proliferation, and they go out and they are defending against any kind of pathogen that is intracellular, that has actually gotten into the cell, or even sometimes to abnormal cells like cancer cells, and they infect them and lyse them. And some of them become memory cytotoxic T cells, again, with that blueprint of that particular antigen that was on the pathogen or the abnormal cell to get ready if it comes back again. So two things that I want to note. One of the important receptors on the helper T cell is called the CD4 receptor. And so sometimes helper T cells are also called CD4 cells after this very important receptor. Cytotoxic T cells have a little bit different receptor on their cell surface over here. Their receptor is called a CD8 receptor, and it's a different protein on the cell surface. And so sometimes cytotoxic T cells, also called killer T cells, also sometimes called CD8 cells. So sorry about that. All right, so let's go back again to these lymphocytes, these B cells and T cells. So remember, they are part of the hemopoietic stem cells that arise in the bone marrow. And these, some of these go to the actual bone marrow. They stay there and they differentiate into B cells. And those B cells are part of our humoral response to the adaptive immune system. Other parts of the stem cells go to the thymus, where they differentiate into T cells. And then these T cells and B cells go to lymphatic tissue, like lymph nodes especially, but also sometimes to the spleen. And that's where they kind of hang out to be ready. So if you remember with the Bozeman video, the introduction to the immune system, he talks about spies as part of the adaptive immune system. This is where the spies would hang out, in your lymph nodes. So let's take a look about what an antigen presenting cell is. So we've talked about this a little bit, but let's look. So let's pretend like we have this bacterium that's a bad bacteria. It's been engulfed by a in this case, a dendritic cell, which is a phagocytizing cell. And so it gets engulfed, and these phagosomes, which is what they call an engulfed particle, gets fused with a lysosome, and the digestive particles break that little guy into pieces. But some of the piece of the original pathogen gets saved, and it gets put on a protein called an MHC2. It's major histocompatibility is what that stands for. 2 protein is kind of like a little cherry on top of a sundae, and it is presented on the outside of this dendritic cell. And that dendritic cell goes scooting off to a helper T cell to activate and call the alarm to the rest of the immune system. So major antigen presenting cells are dendritic cells, as we just saw, macrophages, and also B cells can do that as well. And other cells in the immune system can also act as antigen presenting cells. But this is a very important part of the acquired immune system, where a actual piece of the bad guy is presented on a stalk of a protein that is an antigen that is on the outside of the cell, but it's different every time. Do you see that? Because it's whatever piece of the pathogen, that specific pathogen, is presented on top of that. When we look at the antigen, remember, this is a protein, all right? So this thing would be made of amino acids and synthesized in the cell. The part of the antigen that is super important is the specific, unique part that we call the epitope. So the epitope is kind of the action end of the antigen. And that is the piece that can be variable and different, very different, depending on what the original pathogen was. So let's talk about B cells, all right? So we know that the antigen presenting cells activate the helper T cells. And the helper T cell's job is to then go and activate some B cells in the humoral immune system. So the B cells are very different, okay? You can see that there's lots and lots of different kinds of B cells, each with the capacity to make a certain type of antibody. And it's only the ones that interact with the particular antigen from the invading pathogen that get activated. And then, just like I said before, these certain specific B cells are going to diverge into two populations. One are called plasma cells, and the plasma cells actually are antibody producing factories. They churn out a bunch of antibodies. What's an antibody? It is a protein handcuff, right? So that's what these little guys are. And we call this clonal selection because it's only this particular type of B cell that clones itself to produce a whole bunch of antibodies of the same type in response to this original bad guy up here, all right? The other population of B cells that also go through clonal expansion are called memory cells. They're memory B cells, and they have the blueprint for that to make that particular type of antibody should that bad guy ever come stalking around your body again. So this is humoral immunity. So each B cell has a different antigen receptor, and that antigen receptor is only going to combine with the specific antigen once it gets activated, either by a helper T cell that has had an antigen presenting cell alerted and activated, or by the B cell itself coming into contact with the specific pathogen whose antigen fits with the antibody on that B cell. They become activated, and the B cell then spits out a bunch of cytokines, which are going to activate other B cells that are similar to it, and it also makes the cell go through this clonal expansion. That just means making clones of itself. Some of them are going to be this memory B cell that has that specific blueprint of making that specific kind of antibody. And then other ones are going to make a whole bunch of these plasma cells that begin to churn out the specific antibody for that specific antigen that was on the bad guy. And then after the infection has passed, those plasma cells are going to go through apoptosis and die. But the memory B cells hang around for years and years. So let's talk a little bit about the helper T cell, and again, this active role that it takes in both the humoral and the cell-mediated response. So here I have, this could be, well, this is a macrophage, right? So it's ingesting a bad microbe here. Here is this would be an MHC2 class protein, this self protein that goes on the outside, and then this microbe is digested, but just a little piece of that is put on top of the MHC receptor, and we would call that the epitope, right? So that's the action part of that antigen. And then what happens is the interleukin-1, which is a cytokine, helps to stimulate the helper T cell to be activated. And this is like the secret handshake between this MHC receptor on the outside of the antigen presenting cell and the helper T cells receptor that's called the CD4. Once that secret handshake bonds, that stimulates more interleukin-2 to stimulate more of these helper T cells that have been activated. They go on to then activate B cells for a humoral response to make antibodies in the blood and lymph. And other T cells go on to activate these cytotoxic T cells, which kill the infected cells. So the helper T cell is super important, critical, integral part of both parts of the acquired immune system. So I want to talk a little bit about antibodies. So antibodies are an example of some very important proteins. The antibody shape kind of looks like a Y, and they have heavy chain and light chain proteins. The specific, that part of the antibody is always the same. But what's different is the very tip of it. The very tip of the antibody is the antigen binding site. And that is what is very specific. And so you only have specific antigens that would bind to the antibodies that have this specific antigen binding site that would bind to it. So we have lots and lots of antibodies that are floating around that aren't doing anything right now because there's no antigen that would fit into that antigen binding site just perfectly. Antigens can be variable, right, as we've just talked about. And so the antigenic variation just means that the antibody makes different shapes of this antigen binding site to bind with different types of antigens that it might be exposed to. Remember again that the epitope is kind of the key component to the antigen. That is the piece that is specific to the antibody. So you can see here's the antigen binding site of 3 different antibodies, these blue things. And I can tell right now that this particular epitope is going to fit right in there, right? This one would not. It would not. But this one does bind with this particular antibody here. So your immune system as a vertebrate creates a lot of these different kinds of antibodies just in case a specific kind of antigen comes its way. So what do these antibodies do anyway? I told you they were kind of like protein handcuffs. Well, one of the things that antibodies do is they help in opsonization. So you might remember that word. That's the Greek word that means a tasty side dish, okay? And so what antibodies do is they help to immobilize the invading pathogen to make it easier to be digested by some type of phagocytic cell like a macrophage. And so antibodies help with opsonization. They kind of clump the bad guys together to make it easier for the cells that sweep along and clean them up and kill them to come around. Antibodies are also called immunoglobulins. And sometimes we use capital IG as short for immunoglobulins. So that's kind of the fancy name for an antibody. There are lots of different categories of antibodies or immunoglobulins. You do not have to know those for AP biology, but I just wanted to show you because if you go to medical school or graduate school or take an advanced course in immunology in college, you will learn about these different type of immunoglobulins, and they each have their own specialty. One of the ones that I think is really interesting is this IgA type. And you can see they're all different structures, but they each have an antigen binding site, right? The very tip of that green part would be where it would bind with the antigens. And this is why breastfeeding is so important in humans, because when a mother breastfeeds, she is actually delivering antibodies to her baby through her milk. And that's a really important thing for a newborn whose immune system is not fully functional yet. So let's talk a little bit now about the cell-mediated immune response. So we've talked about, here's a macrophage ingesting an infected some type of pathogen. And so it's going to digest it. It's going to put the epitope on the antigen on the MHC complex here, and that is going to bond with my helper T cell. Remember, this is like the secret handshake with the CD4 receptor. The helper C cell is going to release cytokines that help those helper T cells differentiate into 2 different categories. The memory T cells that are going to remember that one if they ever are exposed to it again, to be able to release an assault faster. And through mitosis, you're going to get a whole bunch of these. Some of the other thing that the helper T cell does is it activates the killer T cells, and those killer T cells are going to identify an infected cell based on this antigen, this MHC complex that is putting up a little piece of the epitope from the pathogen to kind of say, here, in this cell, I have an infection, you know, come kill me, because this cell has been infected and needs to be destroyed. And so that's what the killer T cells do. And it's not just infected cells, it could be abnormal cells as well, like cancer cells. So these cytotoxic or killer T cells are also called CD8 cells because their receptor on the top of their cell is called a CD8 receptor. It's called a CD4 receptor on the helper T cell, just so you know. And so again, here I have an antigen presenting cell. It's going to bond here with an immature cytotoxic T cell, which is going to go through clonal selection. We saw this in a similar way with the helper T cells. Now I've got a whole bunch of these activated killer T cells. Some of them go off to form memory T cells. If they're needed again, they can more quickly get into the active form. And then they go and they would target specific cells, either infected cells or cancer cells, and they would kill them. They would kill them with chemical warfare, like perforin, and they would granulozymes, and they help them, that helps destroy the cell. So here we have this perforin molecule. Once the cytotoxic T cell is going to dock with the infected cell, and it knows it because it's got this CD8 receptor that bonds to this antigen presenting cell, it releases the perforin. The perforin causes holes in the infected cell's membrane, and then that infected cell lyses and dies. It should definitely make sense to you that our immune system would have a way to stop these cell assassins. These cytotoxic killer T cells have got to be stopped because otherwise they could go haywire. So we also have other cells in the immune system called T suppressor cells. And they are exactly what you would think. They say, okay, guys, the infection is over now. We need to slow down and stop going around killing everybody just in case you kill something that you shouldn't. So they would suppress the helper T cells, suppress the B cells from making antibodies, and they would also suppress the macrophages. So what is immunity? So do you remember those memory B cells and memory T cells that we talked about? Those are what we're talking about here. If you are exposed to an antigen the first time you're exposed, your immune response takes a little bit of time to get going, right? So all of those specific pathways we talked about have a little bit of time to get going. But once they get going, they're going to mount a response. And because you're not dead yet, obviously it works, right? And so then things settle down. But if you are exposed to that antigen again, this time because of your memory T cells and your memory B cells, you have a very quick and immediate secondary immune response that you wouldn't even know you were sick because it so quickly comes on and destroys those invading pathogens. But sometimes our immune system reacts to things that it shouldn't. And there are two specific examples of that: allergies and autoimmune diseases. So in an allergy, what happens is that you have these cells that are responding to things that are not pathogens, like pollen. Pollen is not a pathogen. But sometimes, and we don't exactly know all the ins and outs of why this happens. We do know that there is a genetic component, but there are sometimes things that are not pathogens that they sensitize our immune system to make it respond like it is a pathogen. So here is an example of pollen. activating a B cell, which then activates a mast cell. Mast cell is part of our inflammatory response, and that releases histamine. Well, you remember in the inflammatory response, histamine is a good thing because we wanted more blood to come there. We wanted the capillaries to be leaky so our white blood cells can get out into the tissues and fight. But what happens when you have an allergy, an allergic response? Your nose runs, you're sneezing, you're coughing, your eyes run, you feel flushed, you don't feel good. And sometimes it can be worse than that. It can actually affect your respiratory and circulatory system. So that is why we have our immune system is at fault in an allergy, because we have things that are not supposed to be pathogens triggering the release of chemical fighters that are supposed to be reserved for pathogens. And autoimmune diseases would take this to a new level. So we have lots of different types of autoimmune diseases, and we are still trying to find out how this happens and why this happens, because there are millions and millions of people that suffer from these, and it would really be great if we could figure out a way to stop this. Don't have to know all of these. This is just an example. I would know one or two just for a hip pocket example on an AP Bio question, but these are some different autoimmune diseases. And we're not really sure how this all happens. We know that sex hormones seem to play a role in it, as does generally genetic susceptibility. So we can have a predisposition to some of these autoimmune diseases in our genes, and whether females tend to have this more than males. We know that there are external triggers. These can be pathogens like viruses and bacteria, but they also could be drugs. They could be certain chemicals. They could be pollutants. They could be radiation coming from the sun. It could be diet. It could be stress. And then there's some random variables that have to do with just what kind of immunoglobulin kind of collections you have, T cell receptors in your particular body, and factors that are yet unknown. So we don't really know why sometimes our immune system turns on our own cells, but we know that it does. What can we do to help our immune system? Well, moderate exercise definitely improves the immune system. I once read someplace that somebody said, if you could bottle the effects of exercise, it would be the most fantastic drug ever made. So exercise really does a lot of good things. Psychological stress has also been known to disrupt the immune system because it alters your hormones, it alters your nervous system. And we know this, right? When do you get sick? You get sick when you're stressed, right? We all do. And so there is a real physiological reason why that happens. And also, you need enough rest for your immune system to be at its best. So again, when do you get sick? When you're not getting enough sleep. So those are ways that we can help our immune system. So let's talk a little bit about immunity. How can we pass on immunity? So there are two basic ways that we can pass on immunity. This induced immunity means that you can make immunity happen in somebody else. Passive immunity is one type and active immunity is another type. So let's look at passive immunity first. In passive immunity, you are literally taking the antibodies from one organism to another. So the antibodies, remember, are those protein handcuffs. So in natural passive immunity, that would be an example of like breastfeeding when I just said that a mother passes on some antibodies through her milk to her baby. Also, antibodies can cross from the placenta into the fetus before birth. So that's an example of natural passive immunity. Artificial passive immunity is a way that you can take antibodies and inject them into another person. So if you got an anti-tetanus vaccination, you actually got antibodies that were made in a laboratory and injected into you so that you actually have the antibodies to tetanus in your body, which is good, so you won't get tetanus. Active immunity is when you make the antibodies being produced in somebody else's body. So you're not actually transferring the antibodies, but you are making the person's antibodies, making the person's cells produce antibodies. So let's talk about the natural way to do that. Well, the natural way to get a cell to produce antibodies is to be exposed to a pathogen. All right, that's not a very fun way, but that is a natural way. Okay, so if the pathogens get exposed, then you would produce immunity. And back before we had a smallpox vaccination, this is exactly what some people were doing, is they were actually trying to take healthy people because smallpox was so deadly, and they would, go to houses and expose them to somebody that had smallpox. That's a very, very dangerous thing. But the reasoning is, well, then if they survived, if they were healthy to begin with, then they would have immunity then, and they wouldn't be exposed to smallpox when they were sick or not able to be taken care of. So those were some of the drastic steps that we did before we actually had vaccines. But artificial active immunity is a vaccine. All right, so what we do here is that we would take a piece of the pathogen that would not make you sick, but enough so that it would elicit an immune response and your body would produce its own antibodies, your body. So this is an example of like the vaccine for polio. So when you get a vaccine for polio, you are being given broken pieces of the polio virus to which your body would respond and make a bunch of antibodies against polio so you don't get polio. So vaccines are very important. We're going to talk a little bit more about vaccines and about right now as I'm recording this in the middle of COVID pandemic. And so we have some exciting news about some potential vaccines, and there's some very different vaccines that are being developed for COVID as we speak. So this is a good time to pause right before I start talking about the last part of this lecture. Just pause and just make sure that you can answer these questions to yourself or on a piece of paper or maybe with a partner if you are studying with somebody. So I want to talk a little bit about if you were to invent the perfect virus. So if I was going to invent the perfect virus, I would make it infect the very cells that were supposed to lead an attack on other viruses. And I would make this virus mutate easily and often so that it would keep the immune system always a step behind and prevent the development of a vaccine. I would make my perfect virus a provirus. So it would be a RNA virus that would integrate itself into the host DNA and copy itself along with the host. There are sometimes other DNA viruses that just inject their DNA into the host DNA, and it just stays in our DNA. It just kind of stays in there. It's just like a little addition to our genome, and it just kind of hangs out in there for a while, and that's called a provirus. I would make it have a very long latency period so that I wouldn't get sick right away. I could carry it, and I could infect other people without knowing that I was infected. And finally, I would make my virus be transmitted sexually so that shame would be attached to it. And therefore, if people have shame about how they got a particular pathogen, they would be afraid to go get help, and there would be prejudice surrounding that particular virus. Do you know what virus I'm talking about? I'm talking about HIV. HIV has all of those characteristics that I just talked to you about. And we do not have a cure for HIV. We do not have a vaccine for HIV. But thank goodness we have some very good drugs against HIV. And so it is no longer the death sentence that it once was. HIV is called the human immunodeficiency virus. And what this virus does is it infects the helper T cell. That is the host cell. It infects the helper T cell, which is the centerpiece of our acquired immune system. The loss of the helper T cells, because they die, impairs the whole immune response, which leads to AIDS. AIDS is the disease of untreated HIV. AIDS stands for acquired immunosuppressant disease syndrome. And if untreated, HIV leads to AIDS. HIV eludes the immune system because it is, we'll see in a minute, it is an RNA virus. And so it is constantly mutating, and there is a lot of antigenic variation. And so it's very, very difficult for our immune system to latch onto it because it's always changing its stripes. And it can also be a provirus. It can lay dormant in the DNA of the host cell for a really long time. Where did this virus come from, you must say? Well, we have really good molecular evidence that HIV evolved and mutated from SIV. SIV is called simian immunodeficiency virus. This is a virus that affects chimpanzees. And we know that it is very common in Africa. You know, it's kind of, for chimpanzees, it would be like getting a cold almost. But we can trace back, and we'll talk more about this when we talk about the molecular DNA, but we can trace this back, and we know that this area, and we think it was even back in the 1930s, a long time ago, that there was some type of human who was infected with the blood or body fluids of a chimpanzee that was infected with SIV. And in that person, that virus mutated to become the human version of SIV. And that person, we know, traveled along the river, infecting other people through an exchange of body fluids. How do you do an exchange of body fluids? Sexual interaction is one way that that happens. Until finally it got to a large city and it began to spread. And it's a really fascinating history. I just encourage you to read a little bit of the history of how we figured this out, because it is fascinating. We know that in the United States, HIV happened to first infect the gay community. And that gave it a stigma, because at that time, being gay there was a lot more prejudice about being gay. And because it was a sexually transmitted disease, there was a lot of shame associated with it. It is not specifically in Africa, it is a heterosexual transmission. It just happened to be in that particular population when it came to the United States. And so there was a lot of prejudice about that. There was a lot of fear about it, a lot of ignorance about it. And so people were very, very afraid in the 1980s and the 1990s because getting HIV was basically a death sentence, that people were dying of AIDS, and we really couldn't stop it. But then we got some really good drugs, and now through education and through practicing safe sex, we are controlling the HIV transmission a lot better than we used to be. So again, how do you exchange body fluids? Well, unprotected sexual intercourse with an infected partner. This is the major way. Virtual transmission from mother to child. So in utero, during delivery, and by breastfeeding, an infected mother, an HIV positive mother, can infect her baby that way. And then also by sharing hypodermic needles. And we think of this as being something that that drug users, recreational drug users would do, but this is also in poorer countries where sometimes medical equipment is shared and not sterilized correctly. And so that is another way that it's spread. Modes of HIV infection. In males, it is mostly through male to male sexual contact. In females, it is mostly through heterosexual contact. And this is just showing you that specifically who are the biggest populations that is involved with transmission of HIV, and it's young people. And so that's really important to practice safe sex and make sure that you are not exchanging bodily fluids. The main symptoms of AIDS are opportunistic infections that run rampant in an immune system that has been destroyed by the virus. And so a lot of these involve the lung, tuberculosis. There is a particular type of pneumonia called pneumocystic pneumonia that's caused by a fungi that a normal healthy immune system would absolutely destroy, but in somebody with HIV, their immune system is not functioning. Kaposi sarcoma is a type of cancer that, again, a healthy immune system would be able to stamp out, but in somebody with advanced AIDS, they were not able to do that. So what comes in with an HIV virus? What actually comes in? So an HIV virus is very simple. It's got an envelope. It's got a glycoprotein spikes on it. It has inside there is a capsid that actually is going to go into the cell, and it basically has 3 enzymes that come in with it and the piece of the RNA. So the genetic information that gets injected into the cell is RNA. So this is an RNA virus. And so RNA viruses are single-stranded and they mutate a lot. So what are these enzymes? Well, the first enzyme is reverse transcriptase. Reverse transcriptase is going to take viral RNA and recopy it into DNA. We also have the enzyme integrase. Integrase is going to glue the viral DNA into the host DNA to integrate it into the host genome. And that's when we start calling it a provirus. So for a long time, the cell can be harboring this HIV DNA in our own genome, and that's basically the only sign of infection that is there. And then there's another enzyme that's called protease. Protease is an enzyme that kind of edits brand new viral proteins into functioning proteins. Nascent means like newborn. And so as the cell starts to make HIV proteins for a new HIV virus, this protease is needed to make those proteins functional. So let's look at the whole infection here. What is the cell that the HIV virus infects? Doesn't affect your lung cells, doesn't infect your skin cells. It infects your helper T cell. That's the one cell that it infects. So do you remember that CD4 receptor that we talked about on helper T cells? Guess what this HIV virus docks to? The CD4 receptor. So the HIV virus is going to fuse with the CD4 receptor. And so the CD4 receptor then activates and it allows the virus to gain entry into the cell. Kind of uses our own proteins against us, right? And the CD4 receptor. And so what comes in? Well, the RNA comes in, the capsule is going to degrade. I've got this naked RNA, I've got reverse transcriptase, remember that? And that enzyme is going to make a piece of viral DNA from the RNA virus that came in. And now I've got integrase. That's another enzyme that is going to integrate this yellow piece of HIV DNA, viral DNA, into the blue DNA that's my DNA, my host DNA. And so then what happens is that eventually this host DNA would direct the production of new viral RNA and proteins. It doesn't show this here, but the cell machinery would make more viral proteins, HIV proteins. And remember that protease is going to be the enzyme that is going to edit those viral proteins so that they're activated. They bud out from the helper T cell, and now I have a new mature new virion, which is a single virus. So this poor little zombie helper T cell is going to crank out more of those HIV viruses until it dies. And as it dies, it's going to cripple the immune system. So can you imagine the immune system functioning normally when you don't have enough helper T cells? Clearly, that would not be a good thing. And so we can see this happening in this graph. The word titer is sometimes used to represent the concentration of something. So here I'm looking at the CD4 count. Remember, this would be the population of helper T cells. Over here in blue, I'm looking at viral load. That's how many viruses a person has in their body. So let's look what happens here. We have an initial infection. And initially what's going to happen is that the virus is going to start infecting the helper T cells. More and more helper T cells start spitting out new viruses. And so the viral load goes up and the CD4 count goes down. Why? Because they're dying, right? All those viruses are exploding from it and it's dying. But what happens is that our immune system mounts a comeback, and for a while, we keep the virus at bay, right? But that virus continues to just stealthily infect those helper T cells, and this is without treatment. What begins to happen is pretty soon our helper T cells cannot withstand the onslaught of the virus, and the virus continues to increase. Our helper T cells would decrease, and at that point, after a few after years, and again this would be if with no treatment, then at this point AIDS would develop because your immune system is just not there. And at some point a person that was not treated, that was HIV positive, would eventually die based on one of those opportunistic infections with a non-functioning immune system. So what are the drugs that we have? Well, we have reverse transcriptase inhibitors. Isn't that a great idea? We have drugs that prevent reverse transcriptase from doing its job. So it cannot make DNA from RNA. That's a pretty cool thing. We also have protease inhibitors, right? So protease, remember, is the enzyme that edits the brand new viral proteins into functioning proteins. We have a drug that has inhibitors for that enzyme. We have fusion inhibitors, which block the virus from entering the CD4 cell to begin with. We have integrase inhibitors. So remember, integrase is the enzyme that integrates the viral DNA into the host cell DNA. We have inhibitors for that. And what we have found to be very effective is to use multi-drug combinations of 2 or more of these types of drugs into a treatment. And today, because of the use of these drugs, which are very expensive, we have kept people who are HIV positive from developing AIDS. And so millions of lives have been saved. We still do not have a vaccine to prevent it for the reasons that I talked about earlier, and that is that it is an RNA retrovirus that mutates a lot, and the antigens are constantly changing, and so it's hard to make a vaccine against that. But we're still working on it, and we hope to have that come. So this is just a quick diagram. You might want to pause here and just see if you can see all 4 of those drugs that I talked about, where those would come into play. And so what I'd like to do now is I'd like to show you just a quick little video of an HIV animation into a cell.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface, it becomes embedded in the cellular membrane. And at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Awesome. So I hope that was helpful for you, and I will look forward to your questions in class, and I will see you. That ends our lecture part 2 of the immune system.